Dr. Renelle Hendricks is an expert in soil microbiology. In the video, she will be discussing the reliance of plants on microbes for greater productivity and the role Sabel-X Endophytic Trichoderma plays in helping the plant communicate with microbes.
So every part of the plant is actually associated with microbes. You find them below ground in the rhizosphere or the root zone, and you also find them inside the plant tissue, the endosphere. So that is makes up the total plant microbiome, or all the microbes associated with the plant. So I always give this quote just to give you some insight about how complex these microbe associations with the plant is. So the rhizosphere is actually considered one of the most complex ecosystems on Earth. So although we understand quite a bit now, I mean, it's still one of the most complex ecosystems, and the interactions there are really, I think we will learn a lot about it in the next 5 to 10 years. So the rhizosphere, or the root interface, is literally that 5 millimeter of soil surrounding the root. So it's the soil under the direct influence of the root exudates. These exudates are also the food for the microbes living in the rhizosphere. These microbes produce like a slimy polymer we call a biofilm. And the biofilm is there to protect the microbes against desiccation, but also to concentrate all the metabolites and enzymes and to capture all the root exudates. So it's really just like a unique, totally separate environment that is created here because of this biofilm and all the metabolites and microbes and everything living in this space. So as you can see on that picture, the rhizosphere has a unique pH, totally different from the bulk soil. So in this case, it's a maize rhizosphere. So you can see the rhizosphere very alkaline, whereas the soil is around pH 5.5. It's also chemically independent from the bulk soil. So because the rhizosphere is under the influence of these root exudates and all the chemicals secreted by the plant and produced by the microbes, it's a totally different environment from the bulk soil. And it's also crop specific. I even want to go as far and say cultivar specific, because there's subtle differences in the root exudates between cultivars of the same crop. So you can imagine that the types of microbes associated with plants differ with regards to what is available in the rhizosphere. Just to show you, for example, the beans versus maize, the acidic rhizosphere of the beans versus the very alkaline rhizosphere of the maize. Totally different environments. So life in the rhizosphere, it literally established within the first days of a plant's life. So as soon as the rhizosphere becomes active, there's some exudates secreted by the plant. The microbes form the interaction with the rhizosphere, and they form a lifelong relationship to the benefit of the plant, and obviously to their own benefit in the rhizosphere. This community is also very dynamic, so it changes the whole time, and it changes based on the plant's needs. So it's not a fixed community. If you take samples of the rhizosphere, every time you do a sequencing, you see, new microbes, new levels of a specific microbe. The majority of the microbes in the rhizosphere is bacterial. And then we get to the very complex part. This is the communication in the rhizosphere. So there's a three-way communication. First, the plant communicates with the microbes communicate with the plant, and then the microbes are also in communication with each other. This photo just shows how the developing rhizosphere, that first few days when the seed is germinating. So you can see the biofilm holding the soil, that little bit of soil surrounding the root system, and that is where the microbes live. So just to visually show you the communication in the rhizosphere between the plant and the microbes within cells. So if we look at how the plants communicate to these microbes, it's through primary metabolites from plant metabolism, for example, organic acids that the plants produce, carbohydrates from photosynthesis, amino acids, also secondary metabolites like alkaloids, terpenoids, phenolics for example, the flavonoids produced to signal to Rhizophere. So plants use all these metabolites to recruit certain microbes, so it's a signal for the microbes that there's a food source available, to communicate its specific needs, and also to change the rhizobiome based on the plant's needs. For example, when there's stress or when there's a pathogen invasion or whatever. So the plant communicates that by secreting these metabolites into the rhizosphere, and then the microbes can respond to that. And that is why you see the composition of a specific plant's rhizosphere. You find the establishment of a plant-specific microbiome. So how do these microbes communicate back to the plant? So microbes produce hormones, for example, jasmonic acid, ethylene, salicylic acid, growth hormones that induce genes within the plant. So it's systemically taken up into the plant, and it switches on genes in the plant. Some of these genes activate the plant's immune system that we call induced systemic resistance. It improves the plant's resistance towards stress. Then also it's important to know that plants need to distinguish between microbes that are beneficial and microbes that are pathogens. So they use these MAMPs molecules on the surface of microbes. So it's compared to a fingerprint. So this is the MAMPs molecules on the surface of a microbe, or the fingerprints of these microbes. And the plant can, based on that MAMP, it will know this is a beneficial microbe that we can allow into the rhizosphere. We must actually launch like an attack in the rhizosphere and keep this microbe out. And then thirdly, the microbes in communication with each other. So they literally communicate cell to cell with each other. So there's no microbe sitting in the rhizosphere that is on its own mission. I mean, they produce signaling molecules. They communicate to each other what they're doing. They coordinate the biochemical reactions, the production, everything is coordinated. They move in response to each other, that we call chemotaxis. So they regulate the growth, the metabolism, their biochemical processes. Everything is regulated in response to the rest of the team members. So they really work together as a team. It's not one microbe dominating the rhizosphere. It's literally, this is what I contribute, what can you contribute, and they regulate each other based on that. Then you get the endophytes. They are specifically important in this communication between the plant, but also between microbes. So endophytes are very key communicators in this whole system. They literally recruit functional groups. So they will send produce the signaling molecules that will attract nitrogen fixers, phosphate solubilizers, etc., to the rhizosphere, and they work together with these microbes to establish these synergistic interactions with the plant. So just to show you, I mean, if you have a picture of the soil in your mind, this is what it should look like. So you have microbes that are not associated with plants. They're living in the yellow zone, so the bulk soil. They are free living. They are a key part of soil health, suppressing pathogens in the soil, but they have nothing to do with the plants directly. Indirectly, they contribute to plant health, but directly they're not associated with plants, and they don't communicate with these plants. So the moment a plant sends a signal that there's a carbon source available in the form of root exudates, the microbes responding to that signal we call rhizosphere competent. So they establish in the rhizosphere, so they have the unique ability to establish in the rhizosphere and become plant associated. And then the microbes that go one step further, that not only lives in the rhizosphere, but into the plant tissue, living systemically inside the plant, they are the endophytes. So the free-living microbes not associated with the plant are part of the soil food web. The ones associated with the plant are part of the rhizosphere team, or the rhizobiome. And we will quickly look at all the functional groups that have been identified so far. I think there's lots more, but this is what we focus on at the moment. And then the ones entering the plant tissue, the endobiome or the endophytes, we refer to them as the coaches, just based on their key role in communication. So endophytes, as the name says microbes living inside plant tissue. They form this very close synergistic interaction with the plant, and they play a central role in communication or signaling. So they produce these signaling molecules similar to the plant's own signaling molecules, for example, growth hormones. They produce, they can induce genes, they upregulate biochemical pathways, for example, photosynthesis, and they actually participate in the plant's metabolism. So they actively involved in the plant's own metabolism. So they downregulate and upregulate certain metabolic pathways. So it's not just the microbe sitting there and reading signals, it's actually one participating in the plant's own metabolism, switching on genes, upregulating or downregulating metabolism, metabolic pathways. So just a visual of what's happening there. So the endophyte typically, as I mentioned, the coach infecting the plant, becoming part of the plant, systemically contributing to the plant's metabolism, interacting with the plant's genetics, and playing a central role in communication, receiving signals from the plant about the plant's needs and communicating those signals with the microbes in the rhizosphere. So typically the functional groups of microbes that we've so far identified in the rhizosphere are microbes specifically focused to keep insects out. So they produce peptides to keep insects out of the rhizosphere. Microbes that are nitrogen binders, fixing nitrogen and feeding it to the plant. And then the group specifically targeted to keep fungal pathogens out. And you will also always see there's a growth stimulus in a PGPR or a plant growth response also associated with those microbes. Then the group that is targeted to keep bacterial pathogens out, also stimulate growth. And then the group involved in phosphate solubilization. So the phosphates we know, and not only solubilization and uptake of the phosphates, but they are involved in the whole pathway. We talk about the phosphate use efficiency of the plant. So they contribute to the whole pathway where phosphate, inorganic phosphate, is taken up into the plant and converted to ATP, which is the energy molecule inside the plant. So these microbes work together for the good of the plant. They have to keep this plant happy in an optimal state of photosynthesis, and then the plant will actually secrete up to 1/3 of its carbon through the root system to these microbes to feed them. So what is interesting though, we always thought, but okay, 1/3 of the carbon, that's really costly to the plant. I mean, why don't you see a negative, why do we see a positive effect on yield and not a negative if the plant actually secretes 1/3 of its carbon to these microbes? So one of the major functions of the endophytes are to upregulate photosynthesis. And typically they upregulate photosynthesis with about 1/3. So that they actually make allowance for the food for these microbes, for this team in the rhizosphere. So they upregulate, they interact with the plant's genetics, they upregulate the photosynthetic pathways, and that allows enough food to sustain the team in the rhizosphere. So these microbes also know the moment there's a pathogen or there's stress, the first channel that is closed actually, or significantly decreased, is the carbon going out through the root system. So then the plant goes into survival mode, and it must it immediately cuts off the food going out through the root system. So these microbes will do anything to keep this plant out of stress, to ward off pathogens, to protect their carbon source or their food source. So the whole Sabel-X range is based on the principle of these endophytic coaches. So obviously, if you have an endophytic coach, the endophytic coach can signal to rhizosphere competent microbes, and it can put together the best available team. So in soils where there has been a focus on soil health, there's good diversity of microbes, you'll obviously have a better team when these coaches start to signal and put together a team, whereas in soils that are really poor in in microbes that have not been able. So the coach can only put together the team of what is available in the soil. So the endophytic trichoderma strains in the Sabel-X range, they are from the following trichoderma species. So there are strains in the Rhizosphere species, strains in the virulent species, and also the Atraviridae. So in South Africa, we had quite a task to explain to people if it says trichoderma Rhizosphere, it doesn't mean it's free living or endophytic. It's actually the strain code that's going after that. So you can have a free living or a Rhizosphere competent trichoderma Rhizosphere, or you can have an endophytic one. So trichoderma Rhizosphere doesn't mean anything. It doesn't say anything about where, if it's plant associated or free living or endophytic. So it's that strain code that goes at the back. So these endophytes are very aggressive. They literally infect the plant within 48 hours, because the endophytes, your biggest challenge is that just to get them to infect. The moment they infected, they are inside the plant tissue. The plant take care of them. So it's a nice biological to work with, because all the other variables like soil temperature and moisture and conditions outside of your control is irrelevant if a microbe lives inside a plant, because the plant protects it, the plant feeds it. So your only challenge is to get it inside the plant. So obviously, as we discussed, microbes living inside crop plant tissue is crop specific. So it must be able to infect that specific crop and live inside that specific crop's plant tissue. So that's why we do all these infection tests. Typically, the guideline for these endophytes is, if you work with a new crop, you're putting down the coach first. So you have a preventative strategy. You can work at the recommended dose. The moment you work with a crop, an established crop where there's already disease problems, and you want to change the composition of the rhizosphere, you want to get in the right coach, you want this coach to establish a new beneficial rhizosphere community, you go in with a biological warfare approach. And that is typically, the first application is at double dose. So that's just the general guideline.